Hi, welcome to Biology with Miss Hasagawa. So today what we're going to be doing, we're going to be doing a lab with a chicken egg, and we're going to be looking to see what kinds of biomolecules are located in an egg. So we started this unit thinking of what is life, what are the molecules that are needed for life to continue, and what are those characteristics that we consider living. So here we started this whole idea about life looking at a chicken embryo. We're looking at the chicken embryo development, seeing the chick develop within an egg, and it only takes 21s to do that. And all it needs in order to develop is all located inside that egg. So today, we're gonna be looking at, okay, what exactly are the molecules that are located in there that are needed for life? What are those biomolecules and where are they located inside of the egg? In which it only takes 21 days for this cute, adorable little chicken to hatch. So we're gonna be doing a lab. We've got our egg here, we have a hot water bath, we have some sucrose, protein, and starch solutions, and we're gonna be doing a couple of tests. So let's go ahead and get started. So let's go first review the biomolecules. We were talking about lipids before. So lipids, essentially fats or oils. These are hydrophobic. That means that they're a non-polar molecule. That means that they're water-fearing. They repel water. They don't mix well with water, which is really important because they form cell membranes. So your cells, we have trillions of cells within the body, and we're made up of about 60%, depending on our age, of water. And so it would be bad if everything was just kind of all mixed up together. The cells help differentiate different and specific functions, and this cell membrane helps them to do their important functions. And so this is what they look like, kind of like a little bobby pin. And this right here is a cell membrane. Lipids also help the body store energy. They also make these waterproof protective coatings like in the cell membrane. Another biomolecule are proteins, which are one of the most important biomolecule and are a major structural component for living organisms. So these are different pictures of what a protein looks like. This is like a primary protein structure, meaning it's like what it first looks like when it's first made inside of a cell. It's just a chain of something called a monomer form of amino acids. Amino acids is that monomer, that one single molecule that makes it up into a chain. And so then it can get bigger and fold and fold and fold into a really big structure. So proteins essentially are just really important for our structural component because they control cell processes, they help cell transport, so they're essentially kind of responsible for everything that's going on in the cell, and of course fight disease. If they didn't fight disease, well, we did. And so that's what antibodies are. Antibodies are proteins. Carbohydrates. So this right here is a carbohydrate. This particular one that you're looking at is called glycogen. So these provide organisms with energy in the form of sugar, like a simple sugar like glucose. So a simple sugar would be glucose. And then carbohydrates in general, they are a quick source of energy in the body. So that's why if you're gonna go run a marathon, you do like a quick carbo load because you're giving your body quick energy, or you have like those sucrose packets that you use while you're running, and that gives you a quick source of energy. They're also super important for plants because they provide structural support for plants. Plants don't have bones, they have cellulose, which is a carbohydrate. Then we've got biomolecules of nucleic acids. So this is a DNA nucleotide. So this is called a nucleotide, the monomer for DNA, so that one molecule, and a bunch of these nucleotides make a chain. So right here, that's the first thing that they do is store genetic code, DNA. They can also help make proteins, that's something called RNA. And they also are involved with energy transfer with something called ATP. So this is our nucleic acid, our nucleotide for DNA. And again, they make this kind of repeating chain that makes up our double helix of our DNA or our deoxyribose nucleic acid. So that is a very fast review of all of our biomolecules that are important for life. So we're going to be looking at to see which ones are inside the egg. The only one that we're not going to be looking at is nucleic acids, because nucleic acids, they are in DNA, and DNA is going to be everywhere inside of the egg, so we don't really need to look at that. We're going to be looking to see where the proteins, the carbohydrates, simple and complex, as well as lipids are. So follow along with me. First, we're going to do a control. So it's very important to do a control group first, because control groups essentially give you something to compare inside of your study. You're looking to make sure that your equipment is working correctly, and that way you know what a positive result looks like when you do the actual experiment. So we're going to do control to make sure that our lipids, our protein test, our glucose test, and our starch test is working. One of the harder ones to do would be the lipids test, because it's just using a paper bag. And so we're going to be testing that with just some deodorant, because there is oil in deodorant. And so how this works, I'm just rubbing it on the paper bag. And what you should be able to see is it's going to create kind of like a grease spot. So if you can kind of see on the screen that it kind of creates this little grease mark on the paper bag. So if there is a lipid present, it would make something like that. All right, let's move on. Let's start with our glucose test, because we have to heat up our glucose test. And so I've got a glucose solution here, just a glucose tablet mixed with water. I'm gonna put some in my test tube. And then we're gonna test it to see if glucose is in it. Should glucose be in glucose water? I should hope so. So the glucose test is tested with Benedict solution. So let's go ahead and put that in there. It takes 15 drops. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. And then we're going to gently swirl it. I'm going to put it using that, and I'm going to put it in the hot water bath. I'm going to leave it there for about 1 to 3 minutes. The reaction should be pretty quick. Okay, so then let's go ahead and test our proteins. So proteins, we've got protein here. And we're going to put this in a test tube as well. And the protein test, we're testing it with a biuret solution. So we're going to put 10 drops of biuret in the protein to test for protein. So here's the biuret solution. My cap down, so just 10 drops is all needed. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let that mix up a little bit and shake it. As you can see, it kind of changed a little bit color. I think we need a little bit more. Here we go. And swirl it around. So I have a lot of protein solution in there. And as you can see, it kind of changed like a purplish hue. So the glucose test, we're gonna see a grease spot. And our proteins test, we're gonna see kind of like a light purple. Like you see how that's kind of a very light, very faint purple. Let's shake it up again. There we go. Okay. Now we're going to test for, this is just water. Starch. So let me get my starch solution. Put some starch in my test tube. And we're going to test that with iodine. So here is my iodine. I'm going to put that right here. 1, 2, 3, 4, 5, 6, 7. So technically you need 10 drops, but as you can see, we're already getting a pretty big reaction. So it should turn a dark purple, just like it did right there. Now let's go ahead and look at our glucose solution. Look at that. We've got a good color change. So here we've got like about an orangish, kind of a brownish color. So this is what we're looking for when we're doing our lipids, our proteins, and our glucose and our starch test. We are looking for something kind of like a really light purple. So this turned really, really light, but a light purple. A dark purple for our starch test. So there we go. Let's not mix that up. And our glucose test is going to turn a rusty orange, kind of a brownish color. So those are our tests that we're going to be doing. So first up, let's go ahead and do our lipids test. And I'm going to move this over. So here's our eggs and our test tubes. We got egg whites and egg yolks. And I can move this out of the way now. All right. So first up, we have egg white and egg yolk. So let me get a little bit of egg yolk on, or egg white. We'll do egg white first. Smear a little bit on there. And then we're going to do it with our egg yolk. So this test doesn't work all the time. It's not the most scientific test. And it's kind of hard to see like which one leaves like a grease stain. So we'll let that sit and see what we get. But what should end up happening, and we'll see if we get the results, but I'm gonna go ahead and tell you the results. The results should be the egg whites are gonna be test negative for lipids, and our egg yolk will test positive for lipids. So it's really hard, like I can even looking at it here, it's kind of hard to see like which one's like the greasy stain, but it's definitely gonna be the egg yolks. Now let's do our protein test. So our protein test, that's with our biuret solution. So let's get our biuret solution. And let's go ahead and I'll put this out of the way. So we're gonna put about 15 drops of our biuret solution in here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Go. Remember, we're looking for like a light purple. So like how ours turned, like this turned purple, this definitely turned purple. So that means that we definitely have a positive reaction protein in our egg whites. Let's see if our egg yolks have protein. Let's put some of our biuret solution in there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And 10 drops. Put the cap back on. So as you can see with the other one, it was like an immediate reaction. Swirl, 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 swirl. I'm not really seeing any purple. So that is a negative. We don't have any proteins in our egg yolks. Next is our glucose test. So our glucose test, we're gonna test that with our Benedict solution. So let's do our egg whites. So we're gonna put 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 drops in there. Mix it, mix it, mix it. And let's go ahead and put this in our hot water bath, and we'll leave that to rest for a little bit. Let me get another clamp. Now let's test our egg yolk. So let's put about 10 drops in here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I think I did 9. There we go. Mix, 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 mix. Let me mix it nice and good. And I'll clamp the top. So I'm going to be careful since I'm working with hot water. And I'll stick it in here as well. And we'll leave that be for a little bit. There. Okay. So next up, we've got our starch test, which is with the iodine. So let's first do our egg whites. Just need 10 drops. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this one was pretty immediate. We got a really nice dark purple. So this was our positive test. This is our egg yolk. So our egg yolk, it doesn't look like it's changing purple. So that means that it doesn't have any complex carbs in our egg whites, sorry. And then let's test our egg yolk, which I should have said. So here we go, 10 drops. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 drops inside of our egg yolk. Mix, mix, mix. Still again, it's supposed to turn like a dark purple. It didn't turn any shade of purple. So that means it's negative as well. So before I go over the results, let's take a look at our hot water baths, see how they're doing. So this one kind of, let's take a look. This one's kind of clear. It didn't really turn the color I was looking for, but this one definitely did. Because remember, the color we were looking for was about this color. And so this turned this color. This was our egg yolk. Our egg yolk was in here. That means our egg yolk is positive for glucose. And if we look at our egg whites, again, they are negative. So this is the end, this is where we land on it. So this was our lab experiment. Thank you for joining me today. So it looks like when we're looking at our egg that we have egg whites do not have any lipids, but they have proteins. There's no glucose or starch in your egg whites. Your yolks have a lot more. Well, they've got lipids and they have glucose. So our egg yolks only have lipids and glucose, while our egg whites only have proteins. So thank you for joining with me today as we were looking at biomolecules in an egg.